Hello everyone. Today we are going to start with another topic which is Alzheimer's and Parkinson's disease. So both your Alzheimer's and Parkinson's disease are neurodegenerative diseases. So neurodegenerative means there is going to be degeneration or destruction of the neurons taking place. So your most common neurodegenerative disease that is prevalent throughout your world is Alzheimer's disease, and then followed by Parkinson's. So number one is Alzheimer's, second is Parkinson's. So let us start with Alzheimer's. So what is Alzheimer's? Alzheimer's is an irreversible progressive neurodegenerative disorder. So, and in case of Alzheimer's, there is going to be loss of nerve cells in areas of your brain which is going to hamper your memory and learning of a person affected with Alzheimer's. So it is basically characterized by progressive memory impairment, dementia, cognitive dysfunctioning, and may lead to complete vegetative state resulting in early death as well. Okay. So there are three main structural changes that you can see in your brain. Okay. First is diffuse loss of neurons. There is going to be degradation of your neurons. Second is intracellular protein deposit termed as neurofibrillary tangles. So inside your neurons, okay, if this is your neuron, inside the neuron there is going to be deposition of proteins, and these proteins are known as your neurofibrillary tangles, and basically your protein that gets deposited is your tau protein. So there is going to be deposition, intracellular deposition of your tau protein, which makes your neurofibrillary tangles, and lastly there is extracellular deposits of protein which is known as your amyloid beta plaques or your senile plaques. So see, this is the two brains being shown. In your left side there is a healthy brain, and in your right side there is your Alzheimer's brain. So what you see in case of your Alzheimer's is the changes from your normal brain is, first and foremost, the changes that you see is, see your area, okay, your cortex area, it has shrunken, okay, there is shrinkage in your cortex. So basically there is atrophy occurring in your cerebral cortex. Okay, first is characteristics, there is atrophy occurring in your cerebral cortex. So one of your main area of your brain, okay, if you consider this area of your brain is hippocampus, okay, and hippocampus is important in your memory, in storing your memory and learning as well. So generally the type of memory that your hippocampus is associated with is with short term memory. Okay, so in case of your area of your hippocampus, also there is going to be atrophy in your hippocampus, due to which it affects your short-term memory of a person, and as your disease progresses it is going to affect your long-term memories as well. Okay, so first and foremost there was atrophy in your cortical area, second atrophy in your hippocampus. Apart from that, if you see your sulcus and your gyrus, okay, your sulcus they have widened over here, as well as your gyrus they have widened. Okay, so there is going to be widening of your sulcus and your gyrus. So normally this widening occurs due to reduction in your cortical mass. There is reduction in your cortical mass, due to which you see widening of your sulcus and your gyruses. Okay, and lastly, if you see this diagram, see, compare your ventricles, your lateral ventricles, see there is your largening of your ventricles as well. Okay, apart from that, your changes that you see is, so this is your deposition of your extracellular protein, as well as inside your neuron there is accumulation of intracellular proteins known as your neurofibrillary tangles, and your extracellular one is your amyloid plaques. Both your intracellular and your outer cellular deposition, they hamper in your signal conduction through your neurons. Okay, both of them, they hamper your signal conduction through your neurons. So pathogenesis of your Alzheimer's disease. So there is basically two important hypotheses which tries to explain the pathogenesis of your Alzheimer's disease. Those are your amyloid cascade hypothesis and your cholinergic hypothesis. In case of these two, first we'll talk about an amyloid cascade hypothesis. In this case, this basically deals with your intracellular deposition of your neurofibrillary tangles and your extracellular deposition of your amyloid beta plaques. And because of accumulation of these two proteins, what generally happens is there is disruption of your calcium homeostasis, there is increased production of your free radical, excitotoxicity occurs, inflammation occurs, and because of all these factors there is going to be neuronal damage. Whereas in case of your cholinergic hypothesis, okay, when we talk about cholinergic hypothesis, this basically has to deal with your neurotransmitter acetylcholine. Okay, acetylcholine is a neurotransmitter which plays a vital role in memory and cognition of your person. So generally for your cholinergic hypothesis, there is reduction in your choline acetyltransferase. Okay, this enzyme which is your CHAT. Okay, this is choline acetyl choline acetyltransferase. This enzyme is important for your synthesis of your acetylcholine. Okay, and second is reduction in your ACHE, which is acetylcholine esterases. Okay, so they help in your breakdown of your acetylcholine. Okay, so recycling can be done. So because of reduction in both of these enzymes, there is decline in your acetylcholine neurotransmitter, and since there is decline in your acetylcholine neurotransmitter, it impairs your nerve impulse transmission. So due to impairment of your nerve impulse transmission, it also impairs your learning and memory of a person. Okay, so first let us talk about your amyloid beta plaques. So in case one of your major changes that you saw was extracellular accumulation of your amyloid beta plaques in your brain. Okay, and because of your deposition of your amyloid beta plaques outside the cell, it leads to different changes in your brain itself. Okay, so generally this amyloid beta plaques, they are derived from a protein. Okay, so they are derived from a protein known as a amyloid precursor protein, which is present in your membrane, your neuronal membrane. Okay, membrane of your neurons. Okay, so you have your neuronal membrane. In your neuronal membrane you contain your protein which is your APP, which is amyloid precursor protein. Okay, so your basic functioning of your APP, okay, your normal functioning, whenever it is under normal, your main function of your amyloid precursor protein is for your growth. It is used for your growth and repair of your neurons when they get damaged. It helps in your growth and your repair of your neurons. Okay, so but since APP is a protein, after it has been used, it needs to be disintegrated, it has to be recycled. So it can this process of disintegration of your APP can be done by a pair of two enzymes. Okay, basically it can be done by cleaved by two enzymes. So it can be cleaved by a pair of either alpha secretase and beta secretase first, if it is okay. If your APP is cleaved by alpha secretase and gamma secretase, in this case it does not form any amyloid beta plaques. Okay, it is non pathogenic in nature. It does not have any harmful effect. But if your APP is cleaved by beta secretase and gamma secretase, then it leads to deposition of amyloid beta plaques. Okay, again I am telling you, APP is a protein. Okay, its main role is to help in your growth and repair of your neurons. So after its function has been carried out, so just like any protein in your body, these protein also needs to be disintegrated and recycled. Okay, so in order to do that, this APP is cleaved by different enzymes. So if this APP is cleaved by alpha secretase and gamma secretase, then your cleaved product, they do not lead to any pathological conditions. It is normal, so there is no harmful effect coming out from it. But if your but sorry but if your APP is being cleaved by beta secretase and your gamma secretase, then this cleavage will lead to a pathological deposition of unwanted proteins. Okay, so let's see. So this is your APP. I said APP is present on your neuronal membrane. Now its function is to help in your repair and growth of your neurons. So after it has been utilized, it has to be cleaved off. So your cleavage, after cleavage, you can get these two pathways. One is non-amyloidogenic pathway and second one is amyloidogenic pathway. In case of non-amyloidogenic pathway, your protein is cleaved by alpha secretase and gamma secretase. Okay, when it is cleaved by alpha secretase and gamma secretase, your product that you get, which comes out, it is a soluble fragment. It does not accumulate inside your body, so it does not develop any it does not help in development of any pathological condition. Whereas in case of amyloidogenic pathway, what generally happens is this APP is cleaved by beta secretase and it is cleaved by gamma secretase. So on being cleaved by beta secretase, your protein that has been formed, which has been cleaved, left off, they form your amyloid beta plaques. Okay, or you can say they form your, this is a pathological protein. Okay, they form your amyloid beta plaques. So normally under normal condition, whenever this amyloid beta plaques are being formed, they are broken down by microglial cells, microglial cells of your neurons. Okay, or your microglia of your neurons through your process of phagocytosis. Okay, they are normally degraded. Okay, you have your protein amyloid beta plaques being formed, so they are broken down by microglial cells by a process of phagocytosis, as well as you have your enzyme, which is neprilysin. Okay, your enzyme neprilysin, which also helps in your breakdown of your amyloid beta plaques. But whenever your production of your amyloid beta plaque is far more greater, but your destruction of your amyloid beta plaques is less, then it leads to accumulation of amyloid beta monomer. Okay, so first you have amyloid beta plaques being produced. So normally this amyloid beta plaques is broken down by your microglial cells through a process of phagocytosis, as well as by your enzyme neprilysin. So it is going to degrade it. But whenever there is an imbalance between production and degradation, when your production of your amyloid beta plaques increases, whereas side by side there's decrease in your degradation of your amyloid beta plaques, there is going to be accumulation of more amount of amyloid beta plaque. So accumulation of more amount of amyloid beta monomers causes it to aggregate. So they aggregate and then they form your amyloid beta oligomers. So this amyloid beta oligomers, they go and they form your amyloid beta plaques. Okay, your extracellular deposition occurs. So this amyloid beta plaques, they normally, what they do is they initiate your release of your free radicals. They can induce inflammation, correct? They can initiate your cell death process. They can initiate your synaptic dysfunctioning, as well as it can activate your kinase enzyme which is present inside the cell, which can activate your tau phosphorylation, and basically all these factors in the end will lead to neuronal damage. Synaptic dysfunctioning occurs. Okay, synaptic dysfunctioning, why it occurs is, see, this is one neuron, your end of your neuron, this is your dendrite, another neuron. So there's accumulation of amyloid beta plaques taking place over here. So because of your deposition of plaques in your synapses, your signal transduction gets hampered. So that's why there's synaptic dysfunctioning occurring. Okay, so I hope you got it. Monomer, since it is getting accumulated in a large amount, they form amyloid beta oligomers. Plaques accumulation, they aggregate. So normally this plaques, what they do is through other pathways, say inflammatory pathway, your calcium homeostasis imbalance. Okay, so all those factors can damage your neurons, can directly initiate your cell death pathway. Synaptic dysfunctioning, why synaptic dysfunctioning? Again I told you, this is your neurons, this is your, so in your synapse there is accumulation of your senile plaques occurring. So this does not allow your impulse to get carried on from one neuron to another. So there is synaptic dysfunctioning as well. And other than that, there is kinase activation, due to which there is tau phosphorylation, microtubule, which I'll explain it again. Okay, so this is all about your amyloid beta plaque. So next is your we are going to talk about your intracellular deposition, or you can say your neurofibrillary tangles of your tau protein. So tau protein, or tau, is a member of your microtubule associated protein family, and it is a phosphoprotein with multiple phosphorylation site and widely expressed in brain. Okay, so the primary function of your tau is to maintain your microtubule stability. In case of Alzheimer's disease, there is hyperphosphorylation of your tau protein, due to which there is disruption in your microtubules. So I'll explain it in this diagram. So this is your neuron. Okay, so this is your basic neuron, and neurons, they send impulse, right? They may send from stroma to your axon, or from your axon to your stroma. So depending upon from your direction, if it is from axon, from your stroma to your axon, it is known as your introgate transport. So signals going from here to your axon. is a anterograde. If signal is going from your axon to your stroma, it is known as a retrograde transport. So basically, for your signals to travel through your cell with your help of your microtubule, so these are your microtubule aligned with one another. So with your help of your microtubule, your neurons, what they form is they form a network, they form a pathway. Okay, pathway, or you can say this is your cellular highway through which your signals are going to move. Okay, so they microtubules, they come together, okay, and then they form a highway. So through this highway, what can happen is your signals can be transferred. Okay, signals moves through this highway, and generally this microtubule which have come together is held together by a protein. Okay, it is connected by a protein which is known as a tau protein. Okay, it is held together by tau protein, and these tau proteins have numerous sites, which is numerous sites which can phosphorylate your tau proteins. Okay, we told you, right, it has a numerous phosphorylation sites. Okay, so it has numerous sites where which can lead to phosphorylation of your tau proteins. So whenever there is production of your amyloid beta plaque, these amyloid beta plaque, they activate your enzyme kinase. Kinase enzymes get activated. Since amyloid beta plaques are being produced at high amount in case of Alzheimer's disease, these amyloid beta plaques, what they do is they go and they phosphorylate your tau proteins. They are going to go and they are going to phosphorylate your tau proteins. So when your tau proteins, which were holding together your microtubules, they get phosphorylated, they break away from your microtubules. Okay, so so if this is your highway, okay, if this is your microtubules highway and it is being held together by tau protein, tau protein, tau protein, tau protein, tau proteins. Now in case of your Alzheimer's disease, amyloid beta plaques is being formed at more amount. This amyloid beta plaques, they activate an enzyme, they activate kinase enzymes. These kinase enzymes, they go and they phosphorylate your tau proteins. Okay, tau proteins present here, tau proteins present over here. So on phosphorylation of your tau proteins, these tau proteins, they break off from your highway, that is your microtubule highway. So as they break away, when they break away, since they were holding them together, your microtubules also they start to they disintegrate. So thereby there is going to be first and foremost aggregation of tau proteins. After they come out, they are going to aggregate, forming your neurofibrillary tangles, and side by side your highway through which your signals were being conducted or were being passed on also gets disrupted. Okay, so you get reduced you get your reduced signals being transferred through your neurons. So both your, see now this neuron, there is tau getting accumulated, there is senile plaques over here, your dendrites, right? So both your accumulation of tau proteins and your senile plaques, they hamper your signal being transferred from your neurons. And as your main function of your neurons is to transfer signals and they do not do it, they generally die off. Okay, so this is all about your Alzheimer's disease. So next comes your Parkinson's disease. So as I've told you already, Parkinson's disease is also a neurodegenerative disease, and it is the second most common neurodegenerative disease. So Parkinson's is a clinical syndrome with four clinical features. So your basic clinical features that you see in case of Parkinson's disease is, first, that is a bradykinesia. Okay, just like tachycardia and bradycardia, right? Tachy means high, brady means less. So kinesia has to do with movement. Brady means slow. So there is going to be slowness in your movement of a person who is suffering from Parkinson's disease. Second, there is muscle rigidity. Your muscles become rigid in case of patients who are suffering from Parkinson's disease. Third, clinical third cardinal features that you see is resting tumor. Whenever a person is suffering from Parkinson's is at resting, you see his hands is always shaking, his legs is also shaking. That is resting tremors. Okay. Generally, this tremors disappears once you initiate a movement. Okay. Only during resting phase you say shaking of hands and feet or legs during Parkinson's. And lastly, there is impairment of balance leading to disturbance of your gait and falling as well. In case of your Parkinson's disease, since this is also your neurodegenerative disease, your main type of neuron that you lose in case of Parkinson's are your dopaminergic neurons, and these dopaminergic neurons are generally lost from your substantia nigra pars compacta. Okay, as well as there is going to be certain intracellular inclusions known as your Lewy body. One of your most important case of a Parkinson's disease that you see is, again I'm telling you, there is loss of your dopaminergic neurons. Okay, there is loss of dopaminergic neurons, and from which part? It is going to be area from your substantia nigra pars compacta region. Okay, as well as there is intracellular inclusions of your Lewy bodies occurring. So your symptoms of a Parkinson's is generally seen whenever there is destruction of more than 70 to 80% of dopamine containing neurons. Okay, only after there is loss of neurons up to 70 to 80%, and then only you see your symptoms of Parkinson's. So if we come to your pathogenesis, okay, whenever we think about your movement of your body, okay, so first and foremost, your major area of your brain that your Parkinson's disease affects is your basal ganglia. So there is degeneration of neurons occurring in your basal ganglia. Okay, so whenever we think about movement, what we really think is there is motor cortex. Okay, motor cortex sending impulse to your spinal cord, and then spinal cord sending message to your muscles to move. So this is generally a very simple mode of how movement occurs, but this is not how a signal is being transferred from your brain to your muscles for movement. Generally, what happens is you have your brain, that is your motor cortex, it wants to move. Okay, it sends a signal. The signal first goes to your basal ganglia. Basal ganglia checks the signal, sends the signal back to your brain. Okay, telling it to either to move or stop, and then your brain sends it back to your spinal cord, and then it moves on to your muscles. So this is generally what happens, not just directly from your motor cortex to your spinal cord and then to your effector organs. Okay, it is basically motor cortex goes to your basal ganglia, checks it, sends it back to your brain again, and then again the signal from the cortex goes back to your muscles. Okay, this is generally what happens. So basically basal ganglia, okay, whenever you move, your smoothness of your movement, your precision of your movement, it is all controlled by your basal ganglia. So basal ganglia makes all your intricate movements that you intricacy that you see in your movement clear. Okay, so in case, okay, of your movement that occurs, it is, there are two types or two pathways that you see whenever you want to move. So there is direct pathway and there is indirect pathway. Okay, so this one we are going to first talk about direct pathway. So before we talk about it, I'm going to talk about some other things. First, okay, there are few neurotransmitters which are very important. There's glutamate, there's GABA. These two are neurotransmitter. Glutamate is an excitatory neurotransmitter. Whenever we say excitatory neurotransmitter, it means it is positive in nature. Say it will, if someone is doing something, say if you are watching TV and your mother comes and tells you to keep on continue to watch TV, okay, so that is excitatory. Similarly, glutamate will tell your cell to continue to do something, okay, whereas GABA is inhibitory in nature. It will tend to stop whatever you are doing. So if you are watching TV and GABA comes, it will cause you to stop watching TV. So that is the difference between excitatory and inhibitory. Excitatory will further initiate or excitate to keep on doing that particular thing, whereas inhibitory will tell you to stop doing that thing. Okay, so in case of your direct pathway, see over here we have started with your cerebral cortex. Okay, from your cerebral cortex, okay, if you see over here there was cerebral cortex over here. Okay, so cerebral cortex, now you want to move. So generally cortex, from the cortex here movement arises. The cortex is going to send a signal. It sends a signal to your striatum. Now see a connection between your striatum and your SNPR is substantia nigra pars reticulata. Substantia nigra pars reticulata. So this is your substantia nigra pars reticulata. So your cortex is connected to your striatum by your neuron, and this neuron in your terminal is releasing glutamate. So it is, glutamate is getting secreted. So striatum, before I explain it in this, I'll tell you, this is just cerebral cortex. One important thing that you need to know is your this SNPR, substantia nigra pars reticulata, this GPI is globus, globus, globus pallidus, pallidus interna. Okay, so this is substantia nigra pars reticulata, this is globus pallidus interna. So one thing is, your globus pallidus interna and substantia nigra pars reticulata is inhibiting your thalamus. Okay, it tends to inhibit your thalamus. So it is checking your thalamus always. So in case of your direct pathway, what is happening is you have your cortex. Cortex sends a signal to your striatum. Okay, striatum is going again connected to substantia nigra, pars reticulata, or globus pallidus interna with a neuron, and this neuron is secreting GABA. Now see, globus pallidus. See, globus pallidus interna and substantia nigra pars compacta is connected to your thalamus by a neuron, and this neuron is transferring GABA. Now, in case of direct pathway, what is generally happening is because striatum is secreting GABA, okay, your neuron from your striatum is secreting GABA into your substantia nigra, pars reticulata, and globus pallidus interna. I said GABA is inhibitory in nature. Now, GABA is going to inhibit your functioning of your substantia nigra, pars. Again, GABA is an inhibitory neurotransmitter. Since GABA is released, now it is going to inhibit your role of your substantia nigra, pars reticulata, and globus pallidus interna. Now, since this is GABA will inhibit your substantia nigra, pars compacta, and globus pallidus interna, what will happen is your GABA will not get released in your thalamus. So there will be inhibition of your release of your GABA to your thalamus. Due to your inhibition of your release of GABA to your thalamus, what your thalamus generally gets is thalamus becomes active in nature. It gets activated. So generally what was happening? Generally GABA was getting secreted from your substantia nigra, pars compacta, or your globus pallidus interna into your thalamus. Because of secretion of GABA, your thalamus was getting inhibited. Due to inhibition of your thalamus, glutamate was not being secreted back into your cerebral cortex. So in this case, in case of direct pathway, due to your release of your GABA from your striatum into your substantia nigra, pars compacta, pars reticulata, and globus pallidus interna, due to your release of GABA, your release of your GABA from your SNPR and GPI does not occur to your thalamus. So this thalamus gets activated. Due to activation of your thalamus, it releases glutamate back into your, see, it is releasing glutamate back into its cerebral cortex. Since it is releasing glutamate, and glutamate is excitatory in nature, okay, it is excitatory in nature. Because it is excitatory in nature, your cerebral cortex gets excited, and then it sends an impulse to your spinal cord and brainstem to carry on your movement. So direct pathway tells your body to move. Second, you have your indirect pathway. Okay, the same thing is occurring. Okay, if you see, in case of indirect pathway, you have your cerebral cortex over here. Okay, see, there is your cerebral cortex over here. Now in case of indirect pathway, you are sending a signal. Same time you want to send a signal goes one pathway, one same time it is activating both the pathways. One side it is activating direct pathway, and the other side in your striatum, when your signal from your cerebral cortex goes into your striatum, it is releasing glutamate. So the same striatum has neurons which give a rise to both your direct pathway as well as your indirect pathway. So glutamate goes and it activates your striatum. So on activation of your striatum, so generally in this case, okay, if you see, in this case, your globus pallidus interna, okay, this is XE is externa. So whenever I say GE, under normal condition, your globus pallidus externa is controlling your subthalamic nucleus. STN is subthalamic nucleus by releasing GABA. So whenever GABA is released, your STN gets inhibited. Because it gets inhibited, there is no release of glutamate occurring. Okay. So in this case, your striatum from your brain, it releases striatum to your glutamate is released to your striatum. Your striatum, it gets activated, and on activation of your striatum, it is going to release your GABA. Now since GABA is inhibitory in nature, so it goes and it inhibits your global pallidus externa. Now since GABA is inhibitory in nature, what happens? It is going to inhibit your further release of GABA into your subthalamic nucleus. Correct? Under normal condition, this GABA was checking your action of your, was inhibiting your subthalamic nucleus. But now what is happening? Striatum caused release of GABA in your globus pallidus externa, due to which your GABA is not released to your subthalamic nucleus. Since subthalamic nucleus is no longer inhibited, what happens? It is free to act. Now since it is free to act, your subthalamic nucleus, it releases glutamate into your subthalamic nucleus, it releases glutamate into your substantia nigra, pars reticulata and globus pallidus externa, due to which there is activation of your SNPR and GPI. If you see a difference, in case of indirect pathway, in case of direct pathway, GABA was being released and your SNPR was getting inhibited. Okay. In case of indirect pathway, glutamate is being released into your SNPR and it is causing activation of your SNPR. Since SNPR is getting activated, now this pathway is getting activated. So it leads to release of GABA into your thalamus. Again, see, GABA is getting released into your thalamus. Now thalamus is, since GABA is inhibitory in nature, it is going to inhibit your function of your thalamus. It is going to block your action of your thalamus. So when it blocks your action of your thalamus, generally what happens, see at the top, your release of glutamate is inhibited to your cerebral cortex. Okay. So this release of glutamate into your cerebral cortex is getting inhibited. So once your release of glutamate into your cerebral cortex is getting inhibited, you get a signal which tells you not to move. Okay, so side by side, whenever I want to move my hand, say you want to move your hand right now, okay, your brain is going to send a signal to your body to move. But whenever it enters into your striatum, it activates both your direct pathway and it activates your indirect pathway simultaneously. So whenever you want to move one pathway, direct pathway is telling you to move and indirect pathway is telling you to stop. So there is always a clash occurring in your brain itself, which is asking you to move. Same signal is asking you to move through your direct pathway, same signal is asking you to stop through your indirect pathway. So in order to clarify this, in order to ease this out, what happens is there is your substantia nigra pars compacta. Okay, so from your substantia nigra pars compacta, there is one, your dopaminergic neuron arises from your substantia nigra pars compacta, and then this neuron secretes dopamine in your striatum. Okay, so same signal, one signal asking you to move, same signal asking you to don't move. So it becomes very confusing, okay, whether to move or not to move. So this is simplified by an external supply of dopamine. Okay, so your dopamine is supplied from your substantia nigra pars compacta, and this dopamine is released in your striatum. Okay, so since your dopamine is released into your striatum, and dopamine itself is a neurotransmitter. Okay, and this dopamine acts on two different receptors, which is your D1 and the other receptor is D2. D1 is excitatory receptor, D2 is inhibitory receptor. Okay, so your presence of D1 or your D1 receptor is found more in your areas where you have your direct pathway is associated, whereas D2, which is inhibitory receptor, is generally found in more amount in your where your indirect pathway arises from your striatum. Okay, again one signal asking you to one signal asking you to move, that is a direct pathway. The other signal asking you to stop, that is your indirect pathway. So in order to clarify this, what happens is there is your substantia nigra pars compacta. So from your area of your substantia nigra pars compacta, there is your dopaminergic neuron which comes and which terminates in your striatum. So this in your striatum, this neuron, what it does is it releases dopamine. Okay, it is going to release dopamine. So this dopamine has to act on its own receptor. So there is basically two receptors present. One is D1 receptor and D2 receptor. D1 receptor is excitatory receptor, D2 receptor is inhibitory receptor. Now whenever, and this D1 receptor is found abundantly in case of your areas from where your direct pathway arises, whereas D2 receptors are found in your areas from where your indirect pathway arises. Now this dopamine goes and binds to its receptor. So when it goes and binds to your D1 receptor, what is it going to do? It is going to tell your body to move. Okay, tell your body to move, whereas D2 receptor is an inhibitory receptor. I said it is inhibitory receptor. So what is your, and D2 receptor is found in your pathway of your indirect pathway. So indirect pathway was telling you to do what? Was asking you to stop your movement. So when your dopamine goes and binds to your D2 receptor, what does it do? D2 is inhibitory in nature. It is going to inhibit your indirect pathway signaling. So since it is blocking your don't move signal, the only signal that is left for you to do is move signal. So you get a movement. Okay, and then you move by. Whereas lastly, in case of your Parkinson's disease, this substantia nigra pars compacta, which was giving rise to your dopaminergic neuron, is degraded. So since there is degradation of your neuron, your dopamine is not released properly. So since dopamine is not released, your signal from your cerebral cortex, it activates both your direct pathway and it activates your indirect pathway as well. So you get both your signal of your move signal and you get a signal of your don't move signal. So there's a hamper, so it hampers your basic movement. Okay, apart from that, in case of Parkinson's disease, there is also your intracellular protein accumulation of your Lewy body occurs. So generally your Lewy body, it consists of your large your protein, that is your alpha synuclein, and generally your alpha synuclein is a synaptic protein and it is normally present in high amounts in your brain, and it has been seen that your alpha synuclein has some functioning in your signal transduction as well. Under normal condition, your alpha synuclein exists as a alpha helical conformation. But whenever there is genetic mutation, it undergoes conformational change. There is a change from your alpha helical, it becomes your beta sheet structure. it, from alpha there is a change in your structure. Your beta sheet reed structure takes place, which polymerizes, which condenses and forms toxic aggregates as well as amyloid plaques. So these help in degradation of your neurons. Okay, and with this we complete your another topic, that is your Alzheimer's disease and your Parkinson's disease as well.